My name is Ravi Chana. I'm the Executive Director for Diagnostics at ABHI, and thank you for joining us today. It is a pleasure to welcome you to this webinar on quantum technologies and their role in the future of diagnostics, a topic that is rapidly moving from theoretical science into real-world healthcare application. Today's session brings together a set of highly relevant perspectives. We will hear an introduction to the Q-BIOMED Hub, the UK's flagship initiative focused on quantum-enabled healthcare innovation. We will explore quantum sensors for medical imaging, examine quantum-enhanced in vitro diagnostics, and importantly, consider how we move these technologies from lab to clinic. But what do we mean by quantum technologies? At its core, quantum technology harnesses the behavior of matter and energy at the smallest scales. And in diagnostics, this translates into a step change in how we see and understand disease, moving beyond the limits of conventional tools. But why does this matter for diagnostics? Despite major advances in imaging and in vitro diagnostics, we still face fundamental challenges. Many diseases are detected too late when treatment options are limited. Some of the existing technologies can lack sensitivity and specificity, and diagnostic pathways remain resource intensive and hospital dependent. Quantum technology directly addresses these limitations. For example, quantum sensors can detect extremely small biological signals, such as tiny magnetic fields from brain activity or early molecular changes in disease, enabling earlier and more accurate diagnosis. Quantum enhanced imaging offers higher resolution and contrast, potentially revealing disease at a stage that is currently invisible to standard imaging modalities. And emerging applications include non-invasive cancer detection, portable diagnostics, and real-time monitoring, which could fundamentally reshape care pathways. So this is not simply incremental innovation, this is potentially transformational. But why now? The UK is already positioning itself strongly in this space. The Q-BIOMED Hub, which we will hear about shortly, aims to deliver a step change in early diagnosis and treatment, including ultrasensitive blood tests, faster and more accessible imaging, portable diagnostic platforms, all with the goal of early intervention, improved outcomes, and reduced system pressure on the NHS. And this aligns directly with the broader system priorities, particularly the shift towards prevention over late-stage treatment, care closer to home, and then data-driven and precision medicine approaches. However, while the opportunity is clear, the challenge is equally significant. Moving from promising research to clinically adopted scalable solutions will require strong collaboration between industry, academia, and the NHS, clear regulatory and evidence pathways, and a focus on commercialization and adoption, not just innovation. So today's discussions are both timely and important. They will showcase not only the science, but also the pathway to real-world impact, which is ultimately what matters for patients, clinicians, and healthcare systems. I believe this session will provide both insight and inspiration on how quantum technologies can redefine the future of diagnostics. Again, thank you for your time on this webinar. And now I will hand over to our first speaker, Marie-Therese, for an introduction to the Q-BIOMED Hub. Over to you, Marie-Therese.
So thank you, everyone, for joining us. Thanks for ABHI, Ravi and Jonathan for this opportunity. I am the Senior Business Development Manager and here to introduce Q-BIOMED, the UK Quantum Biomedical Sensing Research Hub. We're part of the UK National Quantum Programme, and we are focusing on quantum sensing technologies and how they can be used to improve early diagnosis and treatment. Next slide, please. So here what we will cover, I'll start by introducing the hub, who we are, what we are building, and why it matters for health. Then Professor Jeff Parker will take you inside our quantum-enhanced imaging flagship, including OPM-MEG and quantum sensors for MRI. After that, Dr. Ben Miller will present our in vitro diagnostics platform. And then I will come back to walk you through the commercialization pathway, how these technologies get from the lab to the clinic, and where industry engages at each stage. Next slide, please. Quantum Biomed sits at the intersection of two worlds, quantum sciences and biomedicine, and our job is to bridge them. The hub is a 24 million core UKRI funded hub. Next slide, please. spanning UCL, University of Cambridge, Oxford University, Warwick University, Heriot-Watt University, Cardiff University, and a growing network. Next slide, please. Our leadership team spans UCL, Cambridge, and Oxford, with over 30 co-investigators across physics, biochemistry, and other areas. And we are supported by a clinical steering group that guides our priorities towards the application with the greatest patient impact. And now we're looking to build our industry steering group. Next slide, please. Diagnostic bottleneck. We're all very familiar with this problem. Across the NHS and globally, we're seeing rising healthcare spend, poorer outcome, and increased pressure on diagnostic services. Late diagnosis is a major driver of this. We end up treating patients very late and at a greater cost. So this is the context in which Q-BIOMED exists. We believe there is an urgent need to shift from late-stage intervention to earlier and more precise diagnosis and treatment. Next slide, please. So quantum sensors give us a new toolbox compared to conventional technologies. They can be more sensitive, more precise, smaller, portable, and often less invasive. What's important is what this enables: non-invasive monitoring. Next slide, please. Earlier and more accurate diagnosis, better access to advanced technologies outside specialist center, and earlier new biological insights. Next slide, please. So what actually is a quantum sensor? In simpler terms, these are devices that harness the properties of quantum physics to measure what was previously unmeasurable. Quantum effects like superposition and spin allow sensors to detect magnetic fields, molecular concentrations, and neuronal signals with precision and speed. For healthcare, this translates into three specific capabilities: earlier detection of disease at lower cost, non-invasive brain imaging without the need for expensive cryogenic systems, and rapid point-of-care diagnostics with sensitivity down to single molecules. Next slide, please. There are various modalities within the hub, and these are the two key main technologies: nitrogen vacancy or NV centers in nanodiamond, and Dr. Ben will talk about it in more detail. The second is optically pumped magnetometers, or OPM, which use light and atomic vapors to measure magnetic fields with extreme sensitivity. And you will hear about OPM in Jeff's talk. Next slide, please. Next slide. So our hub is structured around 4 flagships: quantum-enhanced medical imaging, quantum-enhanced in vitro diagnostics, quantum-enhanced interventional tools, and quantum sensors for basic biomedical research. Professor Jeff Parker will take you inside our quantum-enhanced imaging portfolio, and Ben Miller will present our IVD platform. The 2 other flagships we will cover in a future session.
Hello, everyone. I'm Jeff Parker, a professor in the Department of Medical Physics and Biomedical Engineering at UCL. My background is in MRI and the development of various MRI methods. And I'm going to talk to you today about some of the work that we've been doing around quantum sensors for medical imaging. Just as a declaration, really, quantum is nothing new for medical imaging. So since the beginning of medical imaging, 100 years ago or more, there's been a quantum element to the techniques that have been used right back to X-rays. So X-ray production involves the interactions of electrons with atomic electrons to produce the radiation, or at least some of the radiation that generates images. Positron emission tomography, or PET, relies on the annihilations of positrons and electrons in the body to generate photons at 511 keV, which are then used to generate tomographic representations of the distribution of radionuclides within the body. So here we've got an example of a glucose-based radionuclide labeled with fluorine 18, highlighting tumors in the human body. And as a third example, MRI relies on the excitation and relaxation of nuclear states to generate the signal that we use for MR images. So quantum itself isn't anything new for medical imaging, but we're taking a slightly different tack in Q-BIOMED, and the focus of what we're doing is, rather than necessarily inventing brand new imaging modalities, we're exploiting quantum techniques to improve on what's gone before, largely by focusing on quantum sensors. So the specific problem that we're trying to address is that conventional imaging methods such as MRI or MEG, both of which I'll come on to in a bit, generally uses or generates low amplitude signals or non-specific signals. And this means that when we come to scan patients, scans can take a long time. If you've ever had an MRI scan, you will know they can take a long time. It also means that scans are generally expensive and can have low sensitivity. So I'm going to talk about 3 of the themes within Q-BIOMED. There is more, but these are 3 examples that I have. One is work that I'm doing with my team, looking at novel quantum methods to optimize MRI scanning. But I'll also comment on another couple of techniques, one that's looking at developing novel quantum sensors for non-invasive heart monitoring, and another that is looking to detect subtle brain-related signals that are sensitive to injury and disease. And the reason that we're doing this is that by addressing those initial problems with the novel solutions, we can hopefully scan faster, which cuts waiting times. We can improve sensitivity, which means that we can potentially detect diseases like Alzheimer's earlier. And we can generally better characterize and treat diseases in general, but also in this particular set of examples, cardiovascular conditions. And across Q-BIOMED, we're working right from early stage methods where really quite blue sky stuff, all the way through to taking some of the more mature methods that have been developed and applying them in patient groups. So the first example that I'll show you is at the earlier stage of that spectrum in terms of early stage research, and this is the quantum enhanced MRI work that we're currently involved with. So building on the previous slide, one of the challenges that MRI has is that although it's an amazingly successful imaging modality, the signals that we get are generally small and noisy. And this means that we have to scan for a long time to get good quality images. And this is uncomfortable for patients. It means that MRI scans are expensive, and overall it limits throughput, which then has a knock-on effect on waiting lists. And anyone that's ever had an MRI scan might recognize this diagram here. One of the characteristics of MRI is that often when you're being scanned, you will have a closely coupled coil, which is the detector, the current conventional detector that's used in MRI. So these are radio frequency coils, and these are deliberately designed to be as close to the body as possible to pick up signals with maximum sensitivity. And while these work very well, they do have drawbacks. One of the main ones is that these are uncomfortable for patients and they cause claustrophobia. So one of the and also they only enable scanning of a limited field of view. So if, for example, in this case, you'd be scanning the head, but you wouldn't get any signal from the rest of the body. So we're trying to address both of those problems in our MRI theme within Q-BIOMED. This is early stage work, and what we're doing at the moment is we're moving away from that closely coupled coil approach to detecting MRI signals to looking at the far field. In other words, placing detectors at a distance away from the body rather than in close proximity. So the diagram here is a schematic of a real MRI scanner. This happens to be one at the University of Nottingham who we're collaborating with. The scanner is this set of grey boxes in the middle, and the MRI scanner board, the place where the patient lies, is shown in the middle of the scanner. And conventionally, the coils that are used to detect the MRI signal would be in the middle of that scanner board, as shown by this sort of pink square here. So what we're aiming to do is to move those detectors out of the scanner bore into the periphery of the MRI environment, outside of the scanner entirely, potentially. And that then gives us the opportunity not only to be sensing at a distance, but to replicate those sensors within the MR environment in a way that's optimized to detecting signal far off. And these detectors themselves are being developed currently. I can't go into the details of the methods that we're using to develop these because there's quite a lot of IP associated with this, but we're using quantum principles to enable these sensors to maximize sensitivity for these far field detections. And here's an example of some work that's going on at the moment. This is a field map of the emerging signal in terms of the magnetic field from the electromagnetic radiation that emerges from the patient when the imaging takes place. And you can see that there are hotspots of signal outside the scanner ball, which gives us hope that we can identify regions within the far field away from the scanner that we can use to detect the MR signal. So we can use these maps to help inform us where we're going to place these far field detectors to improve signal detection. So as I say, this is a relatively early stage project. We're at the very early stages of that early stage project. So we're currently working on computational simulations. As you can see here, there's also parallel work ongoing in terms of small scale physical prototyping of systems that enable us to characterize and understand what's going on better. And assuming that those two stages are successful, we're going to move on to full scale implementation with a clinical MRI scanner and then test this in humans within the scope of the Q-BIOMED grant. The second area that I want to touch on is using a piece of equipment called optically pumped magnetometers. And there are two contexts that I'm going to describe here. One is applied to the heart, and then I'll follow up with one applied to the brain. So in the cardiac sense, this is looking at a technique used called magnetic induction tomography. So this is a technique that's been around for a while, but it's never really been used successfully in studying human tissue. It's only been used in sort of materials applications, and also, I believe, for identifying landmines of all things. This isn't work that I've done, but this is colleagues at UCL. So the idea behind an optically pumped magnetometer is if you imagine that you've got a gas, a particular type of gas, within a cell, so a small glass box. These little green circles with the arrows on represent the atoms of that gas. If we shine a laser, a polarized laser, onto the gas in this cell, the spins or the atoms within the cell all line up with that laser light that's being shone onto the cell. And if we then expose that cell to some external magnetic field, the alignment of those spins or of those gas atoms is changed by the presence of that external magnetic field. And that external magnetic field may have healthcare relevance. So, for example, it might be a spontaneously emitted magnetic field, as we'll see in the brain example later, or it could be an induced magnetic field in a tissue, which we'll see in the cardiac example in a moment. After the external magnetic field has had this influence on the atoms, that can then be read out by another light source, which can then be detected. So this gives us our mechanism for detecting these magnetic fields. And crucially, this is an extremely sensitive approach to detecting very small magnetic fields and their fluctuations. So the first example of the application of this is in the heart. So this is, again, relatively early stage work, but using an existing technique and bringing it into healthcare applications. And on the right, where the sample is indicated, that's where you would envisage the cardiac tissue to be. The cell that I was referring to earlier with the vapor or the gas in it is this box underneath. And the way that this technique works is that externally applied magnetic or radio frequency fields generate eddy currents within the tissue of interest, which would be the heart in this case. And those eddy currents are dependent upon the conductivity of the tissue that's being imaged. So by exploiting this technique to measure the emerging signals associated with those eddy currents, we can get a handle on the conductivity of the tissue that's being sampled. And by arranging these cells as an array over the patient, in this case over the patient's chest, the hope is that we'll be able to use this as a way of mapping conductivity within the heart, which is important for understanding the functional status of the heart and the signals that are transferred within the heart muscle arrangement. So the last example I'm going to show is again using those principles of OPMs, this time applied in the brain to a technique known as magnetoencephalography. So a very long word, typically truncated to MEG. And this is a technique that looks at the intrinsic magnetic field fluctuations associated with brain function. So this is a technique, again, that's been around for a long time. So MEG was introduced in the 1990s as a technique for measuring these spontaneous fluctuations in the brain due to neuronal firing. And historically, the equipment would look something like this. A patient would have to sit very, very still in this enormous piece of apparatus up here, which was designed to detect these magnetic field fluctuations. And the reason this was so big is that it used superconducting quantum interference devices, another quantum device, which work well, but need a huge amount of cooling in order to be effective. So they have to be cooled to close to absolute zero. So all of this equipment was largely due to that cooling requirement. The big downside of this is that the individual being scanned had to stay very, very still, so no movement allowed, and that really limits patient adoption. It's also very expensive. So the technique that colleagues at UCL and others have developed over the last decade or so is miniaturizing this approach using optical pumped magnetometers, as shown previously. In this case, arranged using a frame like this in an array around the head in order to detect the subtle magnetic field fluctuations associated with brain function. And this has the big benefit of firstly being a lot cheaper than the old style techniques, but also being a lot more patient friendly. So patients can move primarily when they're wearing one of these devices at the bottom, but they can't in the top here. So that opens up a whole new range of clinical questions that can be asked, but also makes it much easier for patients to tolerate. And here's an example. This is in kids. So this is a study where really we're demonstrating, or my colleagues are demonstrating, how user-friendly this is by taking this principle of multiple OPMs arrayed in a different array here. But as you can see here in a pediatric setting, very user-friendly, very amenable to patient use, and generating these fluctuating signals which represent brain function and being able to localize them successfully as well. And this is associated with a lesion which is detected on MRI over here. And just as a final example, this shows the benefit of the approach. Competing techniques, so EEG, which is detecting the electrical component of the same signal effectively rather than the magnetic component, for various reasons is much more susceptible to artifacts. So here we can see a lot of movement-related artifacts associated with muscles and so on, really obscuring what's going on in EEG. The MEG approach, particularly in this more mobile setting, is very competitive with the EEG measurements. As you can see here, the signals are far less corrupted by this noise, which is due to motion and muscle activity. So in summary, we're exploring quite a diverse range of quantum-enabled technologies for imaging, and the focus is on sensors. So we're developing a broad set of sort of next-generation quantum-enabled sensors. And one of the really attractive features of Q-BIOMED is that we're spanning from very early stage research, really quite blue sky, high risk stuff, all the way through to early patient studies demonstrating feasibility of more mature quantum-enabled techniques in patient populations. And that breadth also plays into the number of diseases where we can have an impact. The examples here include neurological conditions and cardiac conditions. But when we're talking about more general techniques such as MRI, that really plays into all of the areas where radiology is conventionally useful. I'll finish there. Thank you very much.
Thank you, Jeff. I think I've agreed with Marie-Therese. We'll come to her presentation at the end. So Ben, are you okay to go next with your presentation, please?
Yep, no problem. Just let me get the slides up. Okay. Can everyone hear me okay?
Yes, we can hear you.
And everyone can see those slides. Okay. Yeah, so I guess Marie-Therese will come to the overall structure of the hub later, but I'm going to present some of our work mostly on Flagship 2 and Flagship 4. So Flagship 2 is our work package on in vitro diagnostics, and Flagship 4 is our work on intracellular sensing. And so, yeah, as you can imagine, these are very closely related activities, both in terms of applications and also in terms of the technologies that we use. So for both of these flagships, we're using NV centers in nanodiamonds, which I'll explain a little bit about now, and then I'll just go on to some of the applications that we've been working on. So yeah, I'll start with what is a nitrogen vacancy sensor. I mean, a little spoiler. What it is a nanoscale optical sensor in diamond, and how it can be used for biosensing, which I would broadly split into two areas. The first is using it for labeling for labeling other biomolecules and generating signal. And the second is for measuring physical properties. So one we're using it as a label that you would sense with by some other means, and in the second we're using it as a sensor itself that you then interrogate with light. And then I'll just chat a little bit about the demonstrations and the current capabilities and future capabilities. So firstly, what is a nitrogen vacancy sensor? So it's an atomic defect in diamond. You can see the diamond lattice here on the left. The green spheres are carbon atoms, and then the N is a substitutional nitrogen for one of the carbons, and the vacancy is a missing carbon. So you have 5 unpaired electrons, or 6 for the negatively charged state, and they hybridize to form something which is equivalent to 2 electrons, and each electron can have spin up or spin down. And so your net spin can either be 0 if you have up and down, it can be 1 if you have up and up, or -1 if you have down and down. And so, yeah, this is the important thing, is that you have this spin defect in diamond, a defect that can take these 3 spin states. The other thing I wanted to highlight on the right is that you can host nitrogen vacancy sensors in many different form factors of diamond, and it's very cheap and well, relatively straightforward, or as in, well, it's commercially done to manufacture diamond and manufacture diamond containing nitrogen vacancy sensors. So by illustration, here is in the middle is here is a diamond chip, which is typically a few millimeters by a few millimeters. Here is an MV center in a sort of AFM style cantilever tip that you can scan over a sample. Here are some nanopillars that you could potentially imagine putting a cell on top and measuring different parts of the cell with these pillars. And then here are nanoparticles, and this is what what almost all of our work is on, so nanoparticles, and the idea obviously behind that is that nanoparticles are very straightforward to integrate into biological systems, and this is routinely done in almost every area of biophysics. Diagnostics uses some kind of nanoparticle label, or nanoparticle or molecular label. So let me just go forward. So I won't go into this energy structure just very much in terms of time, but the important thing to note is that they're fluorescent. So this means between this ground state and this excited state, you can excite with green light and they emit red light. So this is the fluorescence, and this is how you initialize, this is how you sort of initialize your quantum state and how you read it out. And you have this fine structure between the different spin levels. So you have spin 0 and spin +1 and -1 here. And there's this small energy gap between them, which is much smaller than this energy gap. So this energy gap here is in the microwave regime. So what this means is that you can use microwave energy photons, to switch between these spin states and to do your sort of quantum controlling the spin. And then the final point to note is that because of this singlet dark state, when you have, you can read out the spin state. And this is because the plus or -1 states are sort of preferentially coupled via this dark state, which doesn't emit a red photon. And so that means by exciting with green light and then measuring the amount of red light emitted, you can tell if the nitrogen vacancy center is in spin 0 or spin +1 or -1. So these are the sort of the key concepts, and you can actually see that here. So when we apply microwaves to switch the spin state from +1 to -1 using these resonant transitions, much like the energy levels in an atom. You know, the electrons, the electron shells in an atom, it's sort of a similar principle. You have this specific energy gap that you can then address with a photon, in this case, a microwave photon, which is very convenient. So you can see that when you match either this ground state splitting delta E or this excited state splitting delta E star, you have this dip in fluorescence as you're pumping into the + and -1 states. So, to summarize, the important things to remember about nitrogen vacancy sensors is it's a nanoscale defect in diamond. It can be hosted in nanoparticles down to about 10 nanometers. They fluoresce, and their fluorescence emission intensity can be externally controlled. So in a way that I'll explain in a second, this conveys capabilities for labeling for in vitro diagnostics. So if we just simply replace some nanoparticle label, like a gold nanoparticle in an in vitro diagnostic, we can get with a diamond nanoparticle, and we apply these same principles of using it as a fluorescent label, but a fluorescent label whose emission can be controlled. And I'll explain this further in a second, but we can end up getting high sensitivity in the sort of femtomolar regime currently, but potentially getting down to sort of attomolar. It's very chemically and optically stable, amenable to complex samples, and yeah, it can be a drop-in replacement for many assays using straightforward, relatively sort of ubiquitous and low-cost optics and electronics. And just to give some ideas about the potential assays that this could be suitable for, you're looking at, yeah, most of our work has been on lateral flow assays, but there's really any sort of labeling assay. So I'll tell you what we've done on this so far. So what we've done is we've taken a lateral flow test, we've replaced the gold nanoparticles. So got lateral flow tests, as I'm sure you know, are most routinely use gold nanoparticles as labels. And so what we did is we just switched out the gold nanoparticle labels for nanodiamonds, keeping everything else the same. So we still have this binding structure, this sandwich binding structure. And then what we do is we use this resonator here to apply our microwave radiation. And what we can do is we can just turn on and off the microwaves at a set frequency. And in doing so, we can turn the fluorescence up and down. And this will affect only the fluorescence of the nanodiamonds. So the limitation that we're trying to overcome is this variable background fluorescence from our substrate, from our samples, which masks low signals and limit sensitivity. But what we can do is by modulating the fluorescence specifically of the nanodiamonds, by making them flash, we can separate their fluorescence from any other sources of fluorescence in the sample, which will be static, or at least slowly varying. So we can do that in the frequency domain. We do a standard lock-in technique, and we can get very good sensitivity. So you can sort of see here the principle. We apply this microwave field, we turn it on and off, and I don't know if you can see, but hopefully this is a line, an immobilized line of nanodiamonds on a lateral flow test, and you can see it sort of flashing whilst the background remains constant. This is what it looks like in the time domain. And then this is what it looks like in the frequency domain. You have this big peak at the modulation frequency. And that means basically you can separate the nanodiamond fluorescence from any other source of fluorescence. You can separate that signal. And so what that means is that we can detect very, very low particle numbers. We're about 10 to the 5 times more sensitive than gold nanoparticles. And we can detect about 26 particles, which is pretty low in such a sort of straightforward low-cost test. Yeah, we have a patent protecting this technique for diagnostics. In terms of applications, I'll just go through this quickly. We've applied this to the detection of HIV RNA amplicons, getting this sort of low femtomolar regime, which is typical for all the assays we've developed really. We applied this to the single copy detection of HIV RNA, and with a short isothermal amplification step of 10 minutes. And what the sensitivity of the readout means is that we can have this sort of short assay time and still get this single molecule sensitivity. We applied this to the detection of SARS-CoV-2 in wastewater, showing 100% sensitivity and specificity. We did a small clinical study detecting antigen of SARS-CoV-2. I actually think that protein detection is probably a more, well, in my view, and I'd be really interested to hear everyone else's view, but maybe a more suitable application for this kind of technology, because I'm just very impressed with the increasing capabilities of nucleic acid amplification tests. And so I think that, you know, by choosing a protein target where you can't do this kind of equivalent target amplification, I think that's where all of our sensitivity translates directly into clinical benefit. Anyway, in this trial, we used 103 samples, which were sort of chosen so they would match the population viral load distribution, and we showed overall about 87% sensitivity and about 95 in this less than or equal to CT of 30. And you can see all of these sort of middle points. These are patients who are detectable with nanodiamonds, but not with gold nanoparticles. We did a little bit of disease impact modeling and showed that what this translates to in terms of diagnostic advantage is that we could detect the mean viral load patient 2 days earlier on average, and we could detect the 95th percentile lowest viral load patients about 2.8 days earlier than a regular lateral flow test. And we're only about 0.6 days behind qPCR. And yeah, this translates to about 2.2-fold more patients detected on day one of symptom onset, which in a pandemic scenario, at the peak of the Omicron pandemic, would have been about 70,000 patients per day. Okay, that's sort of labeling. And yeah, and we see this being suitable for lots of different labeling, I say different formats, solution-based formats, flow-based formats like we have. And yeah, I'd be really interested to hear, yeah, if anyone has any particular ideas what they think would be good for this technology. Switching tack just for the last few minutes, looking at using nanodimes as sensors. So because your fluorescence intensity is modulated by your spin, you can also use them as sensors to detect anything that will affect the spin or that energy transition between the spins. So that's things like magnetic fields. They're very good nanoscale magnetic field sensors, electric field, temperature, pH, because they'll change the lattice, energies of the lattice, and radicals, which produce these fluctuating magnetic fields. So, I mean, one thing we're interested in on the diagnostic side is trying to expand capabilities of these sort of low-cost diagnostics to include measurements of these physical properties, which is not currently possible by other methods. So also, if you have any ideas for that, I would love to hear what would be good applications for that. One example is detecting ferritin. There's a paper detecting ferritin nanoparticles, which produce fluctuating magnetic fields. Oh yeah, some other work I wanted to mention. We're moving towards making this portable. We have this is about a 900-pound version, and we have a grant right now trying to bring this down to about 200 or 300 pounds, making a sort of more scalable, testable format. So yeah, just to be straightforward about the constraints, you have to be willing to accept some instrumentation. You need a reader to read this out, and there may be some environmental sensitivity, but I actually don't think this is a big issue. Potential use cases, I think, are where you need a result quickly. We're talking 1 or 2 minutes or 5 minutes. You want a very sensitive readout despite fast or low volume samples. Because it removes the background, it's sort of inherently amenable to using complex samples. So yeah, in situations like a GP surgery where or a pharmacy, say, where you want to do minimal sample processing and little user handling, but where you're willing to accept some instrumentation, I think these are the kinds of areas that this could be most impactful. Okay, just briefly at the end, I'm just going to cover some of the sensing modalities that we're covering more in kind of flagship 4. So this is looking at rheology, thermometry, detecting reactive oxygen species, the fluctuating fields from reactive oxygen species, and detecting pH. These are the kinds of things that work by Helen and Knowles at Cambridge is looking into in terms of on the nanoscale, intracellular. So you introduce an nanodiamond into a cell, you track it through the cell, and then you take measurements in different areas of the cells of these different properties, and you do it really, the sensing volume we're talking about is really sort of 40-50 nanometers. So we're talking about very local single cell subcellular measurements. So what they've demonstrated so far, and I'll whiz through this, and I might get some of this wrong, but please follow up with Helena if you're interested, or Adam, in fact. One thing they're looking at the moment is organelle-specific reactive oxygen species sensing during X-ray therapy. So you might be interested in when you're treating a cancer cell, the reactive oxygen species and the DNA damage might give you some indication of the impact on the patient and the impact on the cancer. So I think the way this would work is by taking some of the tumor and then applying radiation in vitro, and then using nanodiamonds to measure spatially the generation of these reactive oxygen species. You do that by relaxometry. So basically what you can do is, as I said, with a laser, you can set up your quantum state into spin 0 or into a known spin state, spin 0 or spin 1. And then you just wait and you time how long it takes for the spin state to reset back to a thermal equilibrium, which will be a mixture of states. And that's what you can see here on the right. You can see gradually over a few hundred microseconds, you're sort of relaxing from this pure state 1 to this mixed state. So this relaxation time will be accelerated, as in it will happen more quickly, if there are these fluctuating external magnetic fields, which these unpaired electrons in reactive oxygen species produce. So they'll have these fluctuating magnetic fields. they increase the rate of this, and so you shift this curve to the left, and that you can measure. As you can see here, the curve is being shifted to the left in a strong response to this MRI contrast agent, which produces a relatively strong magnetic field because it's got this 3+. It's a 3+ ion. So one thing they're looking at is detecting the creation of hydroxyl radicals by this Fenton reaction. So you produce this hydroxyl radical here, and you can see that over time there are changes to the T1 as this reaction progresses. They're also looking at doing this in a slightly different way, so detecting these gadoliniums, but then producing an assay in such a way that the gadolinium ions will start close to the sensor and they'll be cleaved and move away from the nanodiamond in the presence of some other reactive oxygen species, that those reactive oxygen species will react with this linker and displace those gadolinium ions. So this is also, the advantage of that is the gadolinium ions produce a stronger magnetic field than a typical reactive oxygen species, and they're also much more stable, so reactive oxygen species are quite short-lived. They're also looking at a way of doing this spectroscopically. So what I've talked to up until now is non-specific. It will detect any fluctuating magnetic field the same way, so any spin.
It looked like
this ESR
even
approach
your CEA had good results just doing a total cost effectiveness analysis as well. Like
it's spectroscopic, so you can tell which spins you're looking at. And so they're looking to translate that into intracellular sensing of reactive oxygen species. And yeah, my final slide is just on engaging with us. Here are our various contact details, our website. And yeah, thank you very much for listening.
Next slide, please. Now let me show you where our technologies sit on the path to commercialization and what we need from industry at each stage. Most currently, these technologies sit within the first 3 stages of the journey, from early research through to clinical validation. This is where we are actually building, de-risking, and shaping these technologies. At stage 1, TRL 1 to 3, several of our hub technologies are at this stage, including the electromagnetic induction imaging with atomic magnetometer, the quantum sensors for MRI, and other OPM MEG with different applications that Jeff spoke about earlier. So what we need from industry at this stage is research sponsorship, PhD studentship sponsors, and R&D funding partners from MedTech. At stage 2, prototype and validate, TRL 3 to 5. This is a joint effort and where the lateral flow assay platform is, with a patented technology now allowed in the US. And this is also where I want to mention the target product profile, or TPP. We're currently developing the TPP for this platform, defining clinical requirements, use cases, market positioning. And we're not waiting until stage 4 to look at cost benefit. We're already running cost effectiveness and health economic modeling now at stage 2 for both the lateral flow platform and OPM MEG epilepsy system. At stage 3, clinical validation, TRL 6 to 7, both academia and industry lead together. So academic clinicians run the clinical studies and the industry leads on regulatory strategy and packages the evidence for conformity assessment and market certification. So our OPM MEG epilepsy system is approaching this stage, and I wanted to flag something recent and relevant. Last week, the UK government launched the National Quantum Standard Network, a 10 million initiative led by National Physics Lab to coordinate technical standards for quantum technologies. This is the world's first national framework of its kind, which means that the standards being set in the UK will feed directly into global international standards. Next slide, please. There are 2 main routes these technologies take to market: the spin-out model, where a new company is created and holds the IP license from the university, raises its own funding, takes the product forward. But the spin-out doesn't have to do everything itself. It partners normally with an established company, which can bring the QMS, handle regulatory submission, and provide manufacturing capability. So the lateral flow platform and most of our disruptive technologies are heading to this route. On the other side, licensing to an established company where an existing medtech or diagnostics company takes the technology to market using their own regulatory team, QMS, manufacturing, and commercial channels. Our OPM MEG epilepsy work may follow this route depending on the IP position. In the middle, the hybrid model, a spin-out built to a point, then partner with a medtech company. For ABHI members and the wider industry, this hybrid moment is often the commercial sweet spot. Next slide, please. Do we still have some time? I think I can skip this one and move to the next one, please. So this slide gives you a sense of the network we've built: industrial partner, charities, clinical partner, national, international collaborators across our 6 partner university. We're not working in isolation, but this network is not closed. We are actively growing it, and we do need your help and build collaboration with industry. Next slide, please. So how do you get involved? 4 routes: research collaboration, which are joint forces on projects to apply for joint funding as well. So we do have a quarterly translational funding that we run in the hub, and we encourage industrial partners to participate. Industry partnership to explore opportunities to translate the research into real-world application. Clinical engagement to help us bring quantum sensing closer to patient care, and to co-fund the PhD students to invest in the next generation of researchers and build this relationship with a team working on the science that matters to you. This is our website and our email, and we are also on LinkedIn, so please do come talk to us. Thank you very much.
Thank you, Marie-Therese. Okay, we'll open up. We've got about 5 minutes. We'll open up to questions. Feel free to either raise your hand or put something in the chat if you have a question for any of the presenters. While we're waiting for people to raise hands or put questions in, I've got a couple, if that's okay. I think the first one, and maybe this is to all 3 of you, the bit that's important here when we're looking at some of the new technologies coming through is around the cost side of things, because what you want is something that's better from a clinical effectiveness perspective, but also cost effectiveness perspective. I think there's hints in the presentations around this being more cost effective, and Marie-Therese, you mentioned about the health economics piece going on, but is there any more firm indication around what that cost differential is between what quantum provides over existing technologies?
I can probably answer that. So we are working with health economists at the moment, and we're performing health economic modeling for both our lateral flow platform and OPM MEG for epilepsy. So this is running around in parallel of our research. So we're building this value case from the start, and we do have earlier indication as well that these technologies are going to be cost effective in the NHS, because we are also engaging extensively with NICE and with other regulators. Yes, Jeff, and then Ben.
Yeah, I was just going to add to what you were saying there, Marie-Therese. We have a parallel project where we're looking into, as part of other aspects, we're looking into the health economic case for accelerated MRI. So if we can cut the time it takes to do the MRI by 50% or more, what is the financial impact of that to the NHS? So the indications are that there is an economic benefit in scanning faster, but that also needs to be seen alongside the benefits around increasing capacity by reducing the time it takes to do a scan, because you may end up just scanning more individuals and reducing waiting lists. So there's a balance there between the sort of economic case, which is there, but also the broader benefits to society.
Yeah, thank you. Ben?
Yeah, at the risk of preempting all the health economic analysis, I think that one of the things I'm excited about with these quantum technologies is that often what we're talking about is bringing sort of increasing access to diagnostic capabilities that would only be accessible in, say, for example, on the in vitro diagnostics hospital labs, or potentially some of the imaging stuff. And so, yeah, it depends on your comparator. So I would say in terms of straightforward costs, nanodiamonds are very, very cheap. They're just made of any sort of carbon containing material. The primary cost is energy. So places where they have cheap energy can make cheap nanodiamonds. I would say in general, they're sort of comparable or cheaper than gold nanoparticles, for example. So not the primary cost drivers of any of these diagnostic tests. And I would say in terms of the economic return, you're looking not only at like a sort of straightforward cost reduction compared to a lab test, say, but also the additional benefits from identifying diseases early, potentially reducing the cost of treatment and the economic cost of illness.
Okay, thank you. Adam?
Yeah, I was just going to build on what Ben said, which you kind of touched on at the end, as well as the cost differential between the actual things making up the sensors. So I guess nanodiamonds being cheaper than gold nanoparticles. I think it's also the bits of the chain that you can take out by using quantum sensors. So like Ben said with Access, if your GP does have kind of a very cheap in vitro diagnostic device, they can use to get an answer in 5 to 15 minutes that would normally require samples being sent to centralized labs for PCR tests. You obviously no longer shipping samples. You don't have those like expensive thermocyclers and lab equipment. You don't have the dedicated specialist staff that have to run those tests. And then there's also the time thing. So I think we're looking at, as well as the difference in the device cost, it's also those other parts that you're omitting from the chain because you're using the sensor instead.
Yeah, no, thank you. Now, I don't see any other hands up. I'll take. So there's a comment by Mark. It says, on the spin-out model to technology exploitation, gaining investment into medtech is very challenging currently. How are quantum-enabled solutions viewed by investors? Good question. Who would like to?
Yeah, I can answer that. So it's difficult, definitely, especially deep tech and also medtech and diagnostics. But there is lots of effort currently to recognize the quantum sensors technologies. And there is a huge interest from investors as well, not only within the UK, but globally in European countries and the US. There is lots of events happening, lots of investors joining discussions and discussing quantum sensing technologies in that sense, and why they are really useful to invest in for the future.
Okay. Thank you. Conscious of time, we're at time. I don't know if I'll give to Mary-Therese and the team last opportunities to make any comments if you have before we close. So Mary-Therese, over to you, if there's any comments.
No, but I do see a final question actually from Naomi, so she would like to follow. So we'll put you in touch with Ben, I guess.
Yeah, that's fine. Perfect.
And do please reach out to us. Yeah, if there are any questions, we will send a survey feedback.
All right, perfect. Well, Richard, it just leaves me to say thank you very much to the Q-BIOMED team for presenting, and apologies for some of the technical hitches. I blame it on the heat, but my encouragement to everyone, if you'd like to follow up, please do get in touch with Mary-Therese and the team at Q-BIOMED. Thank you, everyone, and have a wonderful rest of the afternoon.
Thanks very much, everyone.
Thank you.
Thanks, everyone. Bye-bye.